Viruses are giant molecules that have the ability to invade and multiply many, many millions of times over within certain specific living cells. And in doing so, of course, they cause disease. They have the ability to take over the building blocks and the energy of the cell in the manufacture of these new viruses. Now, there are three general kinds of viruses, and if you take one of these, let's say poliomyelitis virus, for example, and concentrate and purify it many, many thousands of times over, you can obtain it in crystalline form. If you take one of the poliovirus crystals and slice through it, and then examine the slice by means of the electron microscope, this is what you would see at a magnification of 1,000,000 times. Now, if you take one of those little round particles and enlarge it another 65 times and make a model of it, this is what you would see. This is an example, poliomyelitis, of a virus that causes disease in animals and man. Now, here on the floor is a drawing at the same scale of a virus that causes disease in bacteria. And here we have a model which represents a lot that we know about another kind of a virus, a plant virus. This is a model of tobacco mosaic virus. Now, we show here only 1/10 of the entire molecule of this particular virus. If it were in here, it would be as high as a 6-story building. Now, all of these viruses go only within specific living cells. And when this takes place, they're able to draw upon the energy of the cell and the building blocks that the cell can make to formulate a structure such as you see here, composed of all of these protein subunits and the nucleic acid which is on the interior. Now, it is the latter that we will concentrate on particularly in this program. If we will remove a couple of these protein subunits from this structure, which you can do here, you see on the inside here the little round rod, and this is the rod of nucleic acid. It is this structure, the nucleic acid, which is really the heart of a virus and carries the infective property. Now, my colleague today, Dr. Frankel Conrad, is especially interested in this particular part of the nucleic acid. He studied the tobacco mosaic virus. He was the first person to take a virus apart, tobacco mosaic virus, and put it back together again. He was also the first to show that the heart of a virus, the part that carries the infective property, is the nucleic acid that you see here. Dr. Frankel Conrad.
Thank you, Dr. Stanley. And since we are now supposed to take the virus apart, as a chemist does, I would appreciate if you would give me a hand in taking this model apart so I can illustrate.
I'll be very glad to help you.
Thank you, Dr. Stanley. Here you have now a piece of the virus rod brought down to the size that we can look at it and discuss it better. And as you see now, the nucleic acid is a long thread-like molecule which actually runs for the whole length of the particle, whereas the protein consists of many small separate units, peptide chains, of which actually there are 2,200 in the whole rod that, as Dr. Stanley said, would be a 6-story building. So we have one molecule of nucleic acid and 2,200 molecules of protein combined to form one such particle. In weight, the protein is 95% of the whole material. As you see, most of the stuff is white. But in importance, as you've already heard, the nucleic acid is the predominant component. The nucleic acid can be separated from the virus rod by chemical means, and when this nucleic acid is then applied to a leaf, it will cause the disease exactly the same way as the original virus did. When the protein alone is applied to the leaf of a plant, nothing happens. However, the nucleic acid is not nearly as good an infectious agent as the virus in quantitative terms. It is very inefficient. You have to put about 1,000 times more nucleic acid on leaves to produce the disease. And it was at first thought that this might be due to us chemists having inactivated the nucleic acid, having harmed it in the course of isolating it and separating it from the virus rod. However, soon it became clear that this is not the reason, but that the nucleic acid is so low in infectivity because it is a very delicate chemical molecule. It is a long string-like molecule, as you've seen, and it is composed of many units called nucleotides. 6,500 altogether form one such complete chain. These nucleotides being held together by ester bonds, phosphate ester bonds, which are very susceptible to breakage, both by mechanical agents such as a rapid stirring of the solution, and by chemical agents, acid or alkali, and particularly by enzymes. Enzymes attack this chain very readily and break the long thread into smaller pieces of threads, and it has been clearly shown that only the whole length of nucleic acid, the whole length that goes through one rod, is infectious, and when you cut it in two, you've lost infectivity. It was therefore a very lucky find when we found that we could give the nucleic acid back its original stability by putting it back into protein. It was found that if the protein units were isolated under careful conditions and then brought together with nucleic acid, which also had been carefully isolated so as to minimize breakage, the protein and the nucleic acid would recombine to form once again a virus rod. I can illustrate this here. The protein units have a great tendency to stick together and fit together like this. There must be forces that connect the particular sides in this fashion and prevent them from combining wrongly, such as this. And as they do so, they fit to the next one, and the nucleic acid fits into a groove, holding the whole structure firmly together. This reaction, which we've called reconstitution of a virus, we are going to illustrate in a short film. Here you see those same protein units moving around slowly under Brownian motion at low temperature. But as the thermometer rises now, the motion is faster, and pretty soon some of them will come hit one another, and when they hit one another right, like they now are beginning to do, they stick, like I told you before. Now the nucleic acid comes into the picture, and whenever they have a chance to hit the nucleic acid and the nucleic acid falls into the groove, the motion is accelerated and the completion of the virus particle proceeds at greater rate. The continuing development of this process of particles coming and fitting into the groove leads to one row over the other till we have gradually built up a long virus particle. And you see how the nucleic acid thread is being used up in the process. Less and less of it is hanging down, meaning that it is incorporated into the rod. And when we run out of nucleic acid, then the rod has reached its maximum size and is finished. So therefore, the nucleic acid determines the length of the virus particle. We will now return from the whole virus rod to consider the smaller individual parts. We've now shown you in the film how a virus rod can be formed in a test tube, and probably it is not very different from the way it forms in a plant cell. We've said several times that it is the nucleic acid which is the genetically important part of the virus which causes the disease and determines the nature of the disease. But I would like to now illustrate this to you a little more in more detail, what we mean by this and what we now know about the function of the nucleic acid. The nucleic acid, when nucleic acid from a given strain of virus is allowed to react with protein from the same strain of the virus, you get a virus rod like you've seen. However, experiments have been made in which the nucleic acid from one strain of virus has been combined with the protein from a different strain. And this, we thought, would show us clearly whether the nucleic acid or the protein determines the strain character. You all know that viruses occur in strains that are more virulent or less virulent, and in plants they cause one kind of disease or another kind of disease, and so it is easy to distinguish different virus strains. When we then therefore tried this reconstitution experiment with viruses from two different strains, which is illustrated by this chart, we will call one A and the other strain B. If we were talking of flu, then this could be the regular flu and this could be the Asian flu as an example, but with plant viruses, these are the rods that you've been seeing all along. When we make protein and nucleic acid from strain A, and we make protein and nucleic acid from strain B, and we combine the two so that the nucleic acid comes from B, then in the test tube you will have a mixed virus. You have particles in which the nucleic acid from B was covered with protein from A because that's what we put in the test tube. This mixed virus, when this is put on the cell, gives you pure virus of strain B in the end. The symptoms, the disease, is that characteristic of the parent B, and the virus you can isolate from the plant in the end is also like B. So this experiment clearly showed us that the nucleic acid is boss, and the nucleic acid determines the nature of the progeny from generation to generation. It determines what the virus is like, even when we try to confuse it by wrapping the wrong kind of protein around it. This experiment illustrated to us very forcefully that the nucleic acid is the genetically active material. And we now come and ask ourselves the question, how can the nucleic acid, a long thread molecule like this, how can this have such a specific biological activity? Because this needs much specificity to tell the cell, make virus exactly like that and that kind that I came from originally, and do not make it with protein of the type that I happen to be put together with by the chemists. The information that the nucleic acid contributes to the cell that it invades is immensely detailed and specific, and how can it do that? Well, the nucleic acid, as I mentioned earlier, is composed of nucleotides, of single building units, of which there are four. Four types occur in the cell, four types occur in the nucleic acid, called adenylic acid, guanylic acid, cytidyllic acid, and uridylic acid, which are here only abbreviated with their first four letters. These four differ in chemical structure. You here see a model of one of them in terms of atoms, which are arranged, sugar, phosphorus, and a nitrogen base arranged in a particular pattern which this model illustrates. And there are, as said, 6,500 of them in the long thread of nucleic acid, so that one can see that a good bit of information can be carried, because 6,500 letters can spell out a lot of detail. The fact that there are only four different symbols does not worry us much, because you can remember that the Morse code is composed only of two symbols and a gap, which we might call a third symbol. So there are three symbols in the Morse code, and with the Morse code you can spell, you can write everything that has ever been written or will ever be written. Therefore, the fact that there are only four letters in the nucleic acid alphabet is not of considerable concern to us. However, the question comes, how does this information that is carried here by a sequence of nucleotides, how could it be transformed into actions in the cell, into effects which lead to a production of new material in the cell? This translation of chemical sequence into biological fact is the main key problem that faces biology in general at the moment. Nucleic acids are functioning probably or almost certainly by making the cell make proteins, because everything a cell does is done by the enzymes in the cell, and the enzymes are proteins. It is known that bacteriophage bacteriophage infection causes the production of completely new enzyme proteins in that bacterium, which have not previously been made by that cell. And similarly, probably in other virus infection, new enzymes are produced, which in turn translate the commands of the nucleic acid, make this, make that, stop making this, into biochemical fact. So the nucleic acid information is transferred into protein information. Proteins are being made on the command of the nucleic acid. And proteins, represented here by these white blocks, proteins are consist of 20 different amino acids, which are indicated by numbers here arbitrarily. 20 different types of amino acids make one protein molecule. This protein of tobacco mosaic virus contains only about 14 or 15 of these types, but in general the number is greater. Actually, one virus peptide chain contains 158 amino acids, but these are repetitions of these. There are 15 different ones. In the total chain, there are 158 in one such particle. So we have the problem, how do we translate from 4 symbols to 20 symbols? And there various hypotheses have been suggested, and none of them have been proven definitely right or represent the final word. But one which is easy to illustrate and which is as probably correct as any other that we could now show is that 3 nucleotides together might dictate one particular amino acid. That a group of 3 nucleotides, then there are enough combinations of 3 letters to easily make 20 specific, to give 20 specific variants, and this possibility is now being entertained. So I have here indicated 3 groups of 3 nucleotides, which obviously in actuality are in one long chain and not divided up in groups as I've put them down here, but for the purpose of illustration that might be clear. And now if we say that GAC codes for a particular amino acid that we might call proline, which is, then we put this one on here, PR, which would be number 17 along this numbering system, but we will stick to the particular name here. This might be proline, this might be threonine, this might be alanine. This is all purely speculative. This might be leucine, and now if I'm not mistaken, this is again GAC, and this therefore again proline. We have 2 of the same sequences here and glutamic acid. So you have here a speculative idea of how 3 groups of 3 nucleotides might determine a particular amino acid. Now you could see that if there were 6,500 in a row, they could, in various parts of them, force the cell into making a particular protein with a particular amino acid sequence, one which will be an enzyme necessary for the production of more nucleic acid, another which might be the protein which will ultimately cover the nucleic acid and form the virus rod. But this is only one of the proteins which is being dictated by the nucleic acid in the cell. Now it has recently been possible to chemically alter the nature of the nucleotides. Several reactions have been studied by which you can do this, but one particularly dramatic and exciting case, because it is the simplest in a way, is that you can replace one amino group by a hydroxyl group. This here is a model of cytosine, one of these 4 nucleotides, the one indicated by C, and cytosine has an amino group here. And this amino group, by a chemical reagent, nitrous acid, can be knocked off and replaced by a hydroxyl group. NH2 being replaced by OH. And now you have no longer cytosine, but uracil. And in RNA, this is a very exciting reaction because you've changed one base that originally occurred in the molecule by another base that occurs in this molecule, cytosine to uracil. And so if we take a particular one, GAC, and we say that out of the 6,500 nucleotides in a row, the reagent which we use in very small amounts to achieve minimal effect, the reagent has knocked out this cytosine and replaced it by a uracil, then suddenly this is no longer CAU, but it is now it is no longer CAC, but it's now CAU, and therefore it will code not for proline, but for leucine, because you will find here that GAU codes for leucine. So if this particular change occurred in this particular sequence, it would have changed the protein from a proline in this position to a leucine in this position. I mention this particular case because it has been actually found to occur in recent experiments in our laboratory. We have studied a great variety of mutants produced by such chemical means, and actually it is not easy to produce a mutant in which only one of the bases are changed out of the 6,500. So that the mutant we have particularly focused our attention on was one in which there were about 4 changes in the whole chain of 6,500, but it was a convenient one to isolate and study, and when this was done, it turned out that the protein code of this mutant was different from the protein of the original parent virus. This need not necessarily be so, because, as I say, the nucleic acid is a very long thread, and it commands more than making of more protein. It commands the cell to make enzymes, and this is not an enzyme. This is just the coat protein. So the fact remains that when we happen to study a mutant in which there were 4 changes in the nucleic acid in average, and this is a statistical problem because we don't take one molecule and detect how many changes are on the given molecule. All we know is that there were particles which still retained biological activity, which caused the disease, and the average particle had received 4 chemical changes. When we studied the protein coat of a particle that had been so changed, we found, as I said, that several amino acids had been changed in that protein coat. And to be exact, there were 3. 3 amino acids out of the 158 had been replaced by 3 others. So we here have a exciting fact that the change of one nucleotide in the nucleic acid causes a then self-replicating, irrevocable mutant, so to speak, a step which will continue to reproduce itself in the structure of a protein which we can analyze and locate. Actually, the 3 amino acids that were changed, one of the 3 amino acids that were changed was a change of proline to leucine. That's why I illustrated this particular case here. Proline would have been dictated by this when it had cytosine, and when the cytosine was replaced by uracil, proline was replaced by leucine. And the amino acid sequence of this protein, the whole row of 158 is now pretty well established, and we know that there is a proline near the end of the chain. And we know that the presence of this proline near the end of the chain makes the virus more stable against enzymes than it would otherwise be. Altogether, the virus, when reconstituted, is an extraordinarily stable structure. The nucleic acid is covered inside so that enzymes which attack the nucleic acid can't get at it, and the protein is so closely and tightly packed that also protein-attacking enzymes have trouble getting at the virus when it is a complete rod. In particular, there is a stable group of amino acids on the surface of the virus rod, the outer periphery, in a normal virus. And this particular group of amino acids is so stable to the enzymatic attack because a proline group is near to the end. There is a prolyl alanine threonine sequence, and the proline prevents enzymes from attacking this group. However, when we introduced the change through mutation by nitrous acid, this particular proline near the end was being replaced by leucine. And this is why I indicated to you on this particular block of nucleotides that the change of GAU from GAC to GAU made this coding for leucine where previously proline had been coded. Proline. But now here I notice that there is a small mistake. I hope you caught this too. It is difficult to read them upside down. This should be G. And here we have the same triad UUC as here. So this should be threonine. Now we have them right. So this chemical production of a mutant was actually located in the structure of the virus. This is, however, only the very beginning of a lot of work that is before us and other laboratories who are engaged in the same type of work. We have taken these first small steps in beginning to unravel the code that relates protein structure to nucleic acid structure. Gradually, in the course of many years, the mechanism of genetic transformation, information transfer will become clear. Gradually also, the mechanism by which enzymes operate in the cell is being elucidated by many workers in the field of biochemistry. And when this work on genetic information and the work on enzyme function is being put together, our children or our grandchildren probably will begin to have a clear understanding of the complex matter, of the complex pattern of organization, the cooperation of genetic information and enzyme function, which we call life.
Virus was produced for the National Educational Television and Radio Center by KQED San Francisco, in cooperation with the Virus Laboratory of the University of California at Berkeley. Featured in this program was Dr. Heinz L. Frankel-Conrad, Professor of Virology. The series was made possible by a grant from the National Science Foundation. This is National Educational Television.